Dear students, this is our 11th lecture on bioorganic chemistry, and continuing with mechanism of enzyme action. In this lecture we will see the action of chymotrypsin on different substrates. Chymotrypsin is a protease enzyme which is used for hydrolysis of peptide bonds. So this type of a bond, imidic bond, which is present in polypeptides, is known as peptide bond. And in the presence of chymotrypsin enzyme, this peptide linkage is hydrolyzed to give us the products. And the specific about this enzyme is that this will hydrolyze the peptide bonds which are specifically adjacent to an aromatic amino acid residue, that is tryptophan, phenylalanine or tyrosine. All these, they have a benzene ring or aromatic ring present in the substrate. Only those peptide bonds will be hydrolyzed where these types of amino acid residues, they are present. And for the hydrolysis of these types of peptide bonds, this enzyme has these types of active sites: aspartic acid 102, histidine 95, serine 195 and glycine 193. Now what happens is this is the different active sites of the enzymes. Say this is a strand for the enzyme aspartic acid, histidine, serine and glycine. These three active sites, they are present in this part of the strand, whereas this fourth part is present on this hydrophobic pocket of the enzyme, which is non-polar in nature. So glycine lies in hydrophobic pocket which will bind the polypeptide substrate which is to be hydrolyzed. So this hydrophobic pocket will accommodate this aromatic ring present in the substrate. So that is why the presence of this aromatic ring is compulsory for the hydrolysis of such type of polypeptide substrate. So in the first step, a stable enzyme substrate complex between this aromatic ring being accommodated in the hydrophobic pocket will take place in the first step. And then in the second step, as backbone of the substrate, peptide binds adjacent to the catalytic triad. So these three catalytic active sites, they will interact with the substrate. Aspartic acid 102, histidine 157 and serine 195, they will interact. The amidazole base will abstract this proton from the serine, and this nucleophilic oxygen will attack this carbon, and this carbon oxygen double bond will get converted into a carbon oxygen single bond with negative charge on oxygen, and this negative charge will be neutralized by the presence of this fourth active site in the form of glycine. So a formation of covalent enzyme substrate complex of this nature will be taking place in the third step. So basically, if you see, one active site have got attached to the substrate as a covalent intermediate. So this a covalent bond between substrate and the active site of the enzyme has taken place, and then this will neutralize the negative charge on this oxygen. This active site will play that role, and the histidine part will abstracted this proton from the serine and this new nitrogen will become electrophilic in nature. And in the next step, proton transfer from histidine will take place to the substrate, and we will get this type of tetrahedral component will be formed. Histidine will go back to its original position, and then in the fifth step, carbon nitrogen bond cleavage, this carbon nitrogen bond cleavage will take place, which will complete the hydrolysis of this peptide bond in the fourth step. Then after the hydrolysis of one part, the concerted step in which the covalent enzyme substrate intermediate is formed, over here you can see that in the first step this substrate binding to the pocket has taken place. Then this proton transfer has taken place, which we have already explained. Then this transfer of proton has taken place, and then in the next step this hydrolysis of the carbon nitrogen bond cleavage has taken place. In the next step, one part after hydrolysis will be removed from the reaction site, and this tetrahedral intermediate, which will be a short-lived, will rapidly break down again at this place in the presence of water, and the nucleophilicity of the water will be increased due to the presence of this histidine molecule, which will enhance the nucleophilicity, and this will act as a hydroxide ion, and this hydroxide ion will attack this carbonyl to give us this type of tetrahedral intermediate, and this with the breaking of this carbon oxygen bond will form another part of the hydrolysis product, which will be removed as a final step. So this is the mechanism for hydrolysis of polypeptides, where we are having a aromatic ring in the amino acid residue. Then the conformation of this mechanism, that is can be done by artificial substrate, for example 4-nitrophenyl acetate and related compounds like benzoyl alanine methyl ester or acetophenyl alanine methyl ester or formyl phenyl alanine methyl esters. All these they are related to 4-nitrophenyl acetate. And chymotrypsin, this successfully shows the effect of hydrolysis of artificial substrate. So all these artificial substrates, they hydrolyze under this chymotrypsin mechanism, and in all these cases, if we say in case of enzyme, this is the active site, and we react this with 4-nitrophenyl acetate, this will interact with 4-nitrophenyl acetate, and breaking of this bond will take place, and an enzyme substrate complex of the enzyme with 4-nitrophenyl acetate will be formed, like we have seen in case of the real mechanism of the reaction, and ultimately we will get these products. So on the basis of reaction of artificial substrate also, we can prove that that type of mechanism is taking place. Then the mechanism can also be confirmed by enzyme inhibition reaction. So as we have seen that this is the active site of the enzyme, and if we add DIFP, diisopropyl fluorophosphate, we know that this DIFP is very active. This will react with the serine, H from here and F from here will be removed as HF, and they will form a covalent bond between, and an enzyme substrate covalent complex is formed. So if we use this, the hydrolysis of polypeptide is stopped, because chymotrypsin becomes permanently inactivated in the presence of DIAP. So this also proves that the mechanism in the mechanism active site is the formation of a covalent intermediate between enzyme and the substrate. Thank you very much.